Welcome to the Olink Proteomics Laboratory at the Makerere University Hospital, a state-of-the-art facility that marks a significant milestone in advancing biomarker research in Africa. Powered by cutting-edge technology, the Olink Proteomics platform brings to Makerere University and collaborators a revolutionary approach not only in protein biomarker discovery, but also disease profiling.
Makerere University is committed to achieving her goal of being a research-led and research-intensive university for the purposes of looking for solutions for Uganda's and Africa's development, and particularly in the area of health. We are introducing the Olink laboratory, a state-of-the-art laboratory in the area of medicine. It is going to be the second lab on the African continent. This is extremely important because it is going to help us in research in certain diseases.
This platform is built around a technology called proximity extension assay. This works by using pairs of special antibodies with attached DNA strands. When both antibodies bind to the same protein, their DNA strands come close together, join, and are extended to create a unique DNA code. The unique DNA code is then measured using qPCR or next-generation sequencing. This allows for highly precise and sensitive detection with very low background noise using just 1 microliter of the sample.
As a hospital, we are proud, and Makerere University, being one of the top universities in the world, is happy to be associated with this. We are happy that we have taken the lead. The analysis platform is not only going to enhance the capacity of the hospital to work, because we expect to work with other facilities, other hospitals, both within Uganda and outside Uganda. So basically it means samples can be brought and analyzed from here. The situation has been different. We've been the ones taking samples outside, but this particular platform can enable us to analyze samples within, and we are very happy about that.
The Olink platform allows researchers to measure hundreds of the proteins at the same time across many biological pathways. This helps uncover new insights in systems biology, disease research, and public health monitoring.
We are working with leading institutions around the world to study a number of groundbreaking innovations. 3 examples of what we are working on are: 1, testing bed sensors for remote monitoring of high-risk pregnancies in home settings. Secondly, testing AI algorithms that can estimate gestational age accurately, even for women who present late for their first NC visit. Then thirdly, we are testing novel biomarkers for prediction of pregnancy complications. So to support this, we're able to set up the Olink Proteomics analysis platform here at Makerere University, enabling close collaborations with institutions or with the universities of Cambridge and Oxford in the UK. Together, we aim to identify blood biomarkers that can predict complications such as stillbirths, fetal growth restriction, preeclampsia, and gestational diabetes mellitus.
Our goal was clear: to create a laboratory where Ugandan scientists, doctors, students, and researchers across Africa would study diseases at a molecular level, discover early biomarkers, and develop solutions that are tailor-made for our communities. With the Olink platform, researchers can now analyze thousands of proteins simultaneously gaining unprecedented insight into the biology of diseases such as cancer, HIV, TB, malaria, cardiovascular diseases, metabolic syndromes, the emerging infectious diseases.
Behind the science is a dedicated team of scientists and lab professionals who are trained to ensure the integrity and efficiency of every sample run.
Olink utilizes proximity extension assay, which involves three main steps. That is the incubation step, extension step, and the detection step. During the incubation step, antibody pairs tagged with unique DNA sequences are introduced into the samples. And the overnight incubation, these antibodies specifically bind to the target proteins within the sample within 16-22 hours at 4 degrees in the dark.
I'll take you through step 2, which is the extension and amplification step. This step takes place the following day in the morning, and we use the incubation plate that has been incubating for several hours overnight. During this step, unique DNA reporter sequences are generated for different target proteins and pre-amplified using regular PCR.
In the Olink analysis workflow, the detection step is a critical final phase where presence and relative quantity of target proteins are measured. This step quantifies the DNA reporters formed after specific antibody binding for each biomarker using high-throughput real-time qPCR on the Olink signature Q100 system. And this forms the foundation for the highly multiplexed, sensitive, and specific protein profiling that is enabled by Olink.
After the team has done their work, the laboratory team, all the data that is processed ends up on my desk, where I run and get for you the proteins that are highly predictive of our outcome, all the proteins that are highly expressed in the data. From there, we can run AI and machine learning models to determine the prediction powers of the proteins that are highly significant to determine or to predict the outcome of interest.
From discovery to diagnostics, the Olink Proteomics Lab is a cornerstone in building a healthier future, empowering African scientists to take lead in solving the continent's health challenges.
Given that we are the only ones that are going to have this lab, not just in Uganda, but in a very large part of Africa, I would like to encourage all our collaborators, the public in Uganda and even beyond, to know that we now have this facility and that they can make use of it.
This is not just a lab, it's a gateway into innovation, collaboration, and significant impact to Uganda, Africa, and globally.